My name is Dr. Mark Williams and I'm based in the School of Health and Life Sciences and more specifically in the Department of Biological and Biomedical Sciences. And my research group is within the larger research group of molecular mechanisms and of long-term conditions, and essentially that is part of the newly launched Research Centre for Health, or REACH. And the main aim of REACH is to essentially make a significant contribution to the United Nations Sustainable Development Goal 3, which is aimed at improving the health and well-being of individuals at all ages in society. So just a bit of background on my research career so far. So I have been about a bit. So essentially, I started my undergraduate degree at the University of Strathclyde, where I undertook my undergraduate in immunology and pharmacology. I then moved to the Queen's University in Belfast, where I undertook a PhD in immunobiology. And then I moved back to Glasgow, undertaking 2 postdoctoral research positions, really focusing on blood cancers. And then I moved to GCU 4 years ago to set up my independent research group. And my independent research group is now focusing on 2 major blood cancers. So although they're named blood cancers, they do originate within the bone marrow. And essentially, these 2 blood cancers are associated with a poor clinical course, and that is the blood cancer acute myeloid leukemia and also the other blood cancer, multiple myeloma. And essentially, my main research studies are focusing on how leukemic cells that originate from the bone marrow interact with other bone marrow dwelling cells, such as supportive fibroblasts, which Miss Miari will talk to you about, a PhD student in the lab. And also immune cells such as macrophages can interact with those leukemic cells, and this can drive therapy resistance, which is a major contributor towards poor clinical outcomes in AML patients, essentially. In terms of multiple myeloma, I'm not going to discuss that in any great detail, as my collaborators, both at the University of Strathclyde and also at the Brixton Naval, are helping me undertake collaborative research studies where we're using a combination of computer modelling with biological experiments to generate a new and effective therapeutic antibody for multiple myeloma patients. So in terms of AML, as I said, we are focusing on how leukemic cells interact with bone marrow dwelling cells such as fibroblasts and macrophages. And my group are particularly focused on how both cell-to-cell contact between leukemic cells and fibroblasts or macrophages, and also how factors released by these bone marrow dwelling cells can interact with leukemic cells, drive therapy resistance, and that's through upregulating pro-survival pathways and also proteins such as myeloid cell leukemia 1. So MCL1 is an important target, and you'll hear how basically we're using drugs to target that protein as a strategy to overcome therapy resistance. So one piece of kit which is really important that we use really on a weekly basis, so myself and Katie Miari use it, is the flow cytometer. And with the help of funding from Tenerife Scotland, who've been very generous and funded various research projects at GCU, and also with match funding from the Institute itself, GCU itself, we've been able to purchase this machine. Which, as you can see, it's a nice desktop machine, very colourful, but it's a very powerful instrument which allows us to undertake really detailed analyses of cell behaviours, and specifically we can look at really studying how certain drugs can induce cell death in leukemic cells. And so we can test novel drug combinations, and we can test to see if these unique drug combinations can overcome therapy resistance, which is obviously very crucial. So I'm literally just going to wrap it up there, because in my next video I'm going to show you some data that we've generated from the flow cytometer, and I'll talk you through that. So thank you for listening to this recording, and I hope you enjoy the rest of the workshop presentation. Thanks.
Thank you for staying tuned to our workshop presentation. So I had previously shown you the flow cytometer, that powerful instrument or platform which allows us to assess various cell behaviours, such as the ability of drugs to kill leukemic cells, so cell death, or to assess the impact of drugs on cell growth and proliferation. And now what I'd like to show you is raw data, which has been generated by the flow cytometer, and how we can use the flow cytometer in combination with various assays for assessing cell death to investigate novel drug combinations to overcome therapy resistance and are effective potentially in leukemic patients. So essentially here, I'm showing raw data which has been generated by the flow cytometer. And flow cytometry here has been used in combination with an assay which will detect cell death. And that assay is based on leukemic cells expressing a marker of cell death on their surface. So we use reagents which can bind to that marker of cell death, and we use it in combination with flow cytometry to assess cells that are alive or healthy, as you can see here in quadrant 4 or gate 4, and cells which are undergoing cell death and are at different stages of cell death. And that's due to the leukemic cells expressing this marker of cell death. So essentially, this powerful combination of this assay and flow cytometry will allow us to test novel drug combinations. And essentially, what we can see here is we have leukemic cells which have been grown under normal cell culture conditions and have not been exposed to a drug. And as you would expect, the majority of the leukemic cells are healthy, they're surviving, and over 90% are healthy. However, we would also expect a small percentage of leukemic cells which are undergoing cell death, and those are represented by cells that are in the Q3, Q2, and Q1 quadrants or gates. So essentially, through acquisition of non-commercially available inhibitors from my biopharmaceutical collaborators, we are testing a drug, which I'll name here drug A, on its ability to kill leukemic cells. And as you can see here in this plot, essentially drug A, when it's exposed to leukemic cells grown under normal conditions, so cultivated under normal cell culture conditions, what we can see here is drug A is very effective at killing the leukemic cells. So over 50% of the leukemic cells are dead, as you can see here. However, my research group are focused on understanding how bone marrow dwelling cells or resident cells, such as fibroblasts, which are a supportive cell type, can drive resistance to novel therapies. And essentially what we can see here is that factors released by fibroblasts, when they're grown with leukemic cells which are cultured with these release factors and normal media, what we can see here is that the fibroblast secreted factors are protecting the leukemic cells from the killing effects of this novel targeted drug. So you can see that almost 65% of leukemic cells are surviving and are healthy. So through my research and work that Katie Miari has been undertaking, we know that these fibroblast release factors, these proteins, for example, which are released by the fibroblasts, can activate particular pro-survival pathways and proteins in leukemic cells. So we have then predicted that if we use inhibitors, drug B here, to target those pro-survival pathways, we can resensitize the leukemic cells to the killing effects of drug A. So interestingly, we've looked at this novel drug combination and its killing effects on leukemic cells under normal culture conditions. And as we can see, this is a very effective drug combination, as it's able to induce the killing of over 99% of leukemic cells. Now, obviously, our group is interested, though, in how novel drug combinations can overcome therapy resistance in the context of bone marrow-mediated protection, therapy protection. And essentially what we've done here is we've took leukemic cells, we have grown them under culture conditions where the culture media contains these fibroblast factors. And what we can see here very nicely is that the drug B is able to essentially combine with drug A and induce substantial cell killing. So drug B in this context is resensitizing the leukemic cells to the killing effects of drug A. So we can see here that only about 5% of leukemic cells are surviving now. So that's a massive decrease from 65 down to 5. So essentially, we're getting substantial cell kill. So what does this all mean? Essentially, this novel drug combination we will now test in other leukemic cells from different patients, and also we'll need to test this drug combination in animal models of leukemia. And then essentially this novel drug combination could enter early phase clinical trials and possibly in the future be approved for treatment of acute myeloid leukemia to overcome therapy resistance, and also representing an effective therapeutic strategy for AML patients to hopefully improve clinical outcomes in these patients. So thank you very much for listening to my presentations.
I will briefly cover why modelling of the bone marrow microenvironment is important in our research and how we utilise this to study mechanisms of therapy resistance in the AML cells. Although AML is a blood cancer, it actually originates in the bone marrow, located in large bones of your rib cage, pelvis, hip and thigh, and this is where blood production takes place. On average, an adult will produce approximately 500 billion blood cells daily. This is a huge number of cells, so you can see why it is important for the conditions within the bone marrow to be maintained in order to support these processes. When something goes wrong, this can lead to disorders of the hematopoietic system, and AML is one of these disorders. Panel A depicts a healthy bone marrow where specialised cell types called fibroblasts interact with and support the development of blood precursors so they can mature into fully functional blood cells. Of course, things won't actually be this simple in the bone marrow, as there are a large number of different cell types present along with the bone forming cells. But for today's presentation, I will be focusing on these fibroblasts. In panel B, you can see that along with a limited number of healthy blood precursors, we now also have leukemic cells. When leukemia develops, a number of changes in the bone marrow accompany disease establishment. Fibroblasts adopt different characteristics and transform into cancer-associated fibroblasts, or CAFs, that will support the leukemia cells rather than the blood precursors. Ultimately, the result is a reduction in healthy blood production and an increase in the AML cells. Cells are highly dynamic, and these cancer-associated fibroblasts can interact directly with the AML cells by cell-to-cell contact, or they may also release a number of molecules or factors into the bone marrow called fibroblast-secreted factors. So why are we interested in these interactions between AML cells and fibroblasts? This is because AML cells frequently develop resistance to cancer therapies, which I will refer to as therapy resistance. This is exactly as it sounds. They display reduced or limited response to therapies, meaning that the AML cells can resist the killing effects of cancer drugs and are able to remain in the bone marrow following treatment and survive quite happily. With disease persistence, it is much more difficult to treat and cure the patients. Resistance can be intrinsic or internal to the AML cells, and this is mediated by mutations, for example, that are present in the DNA of these cells. An example would be the presence of protein pumps or transporters on the surface of the AML cells that, when found in high numbers, are able to excrete or pump the drug out of the cell into the environment. Interestingly, resistance may also be extrinsic or acquired. This means that the bone marrow microenvironment and the various cells and molecules present here are able to protect the AML cells against the killing effects of therapy. This is exactly what we are focusing on in the context of fibroblasts. But how do we study such interactions? We are utilising a three-dimensional co-culture system with our collaborator Dr. Monica Guzman to study the interactions between fibroblasts and AML cells. In the image here, you can see the HS5 fibroblasts obtained from the bone marrow tagged with a green fluorescent protein, GFP, so they can be distinguished from the U937 AML cells. When cultured under appropriate conditions, the two cell types will come together into a compact cluster of cells, as you can see in the image, with the fibroblasts in the core forming what we call a spheroid surrounded by the AML cells. By using a 3D model system like this, we can more accurately mimic internal bone marrow conditions in the lab, and therefore we can explore mechanisms by which fibroblasts interact with AML cells to protect them against the effects of therapy and provide therapy resistance. This system reflects low oxygen levels found in central regions of the bone marrow, and these low oxygen levels are known to drive therapy resistance. This is a truly amazing model system to work with, and we hope that in the long term, this will allow us to generate more new therapies for AML and other blood cancers, such as multiple myeloma, that are able to overcome therapy resistance and will improve clinical outcomes in blood cancer patients.